Afternoon to everyone. Welcome to our next session of the webinar CU Talent. Today is the topic of the blue growth, and I would like to give a floor to our coordinator, main coordinator of our research project, to Laura Martin. So please, Laura Martin.
Thank you very much, Maria. Good afternoon, all of you. Good afternoon to the people attending the webinar and our dear speakers. Today we are going to talk about blue growth, sustainable blue growth. As you know, CU Talent, it's an initiative in the research project that involves, between other things, it involves webinar for junior researchers. We really support this initiative, since we think that this webinar dealing with important research issues nowadays could create networking between junior researchers of the different universities. Today, as I said, the webinar deals with blue growth, with sustainable blue growth. Is a couple of words, blue growth, that we can hear very usually lately. They are dealing with economy, they are dealing with resources, they are dealing with well-being. If I had to make a small resume of what sustainable blue growth could be, I would say that it is itself a long-term strategy to support sustainable growth in the marine, and we cannot forget the maritime sector. So it sees, or they say that sustainable blue growth faces seas and oceans as drivers for European economy, for world economy, and have a great potential, seas and oceans, for innovation and growth. So I would say that it's a long-term strategy, a sustainable strategy, where research is very important. It plays a very important role, because it's what is going to provide us the tool for the sea and the ocean to have the enough potential for innovation and for growth. Okay, so today we have two speakers. We have two speakers coming from different universities of our alliance. We have Trinetta Mukherjee. Did I say it in the right way?
It's Mukherjee.
Mukherjee, okay. Sorry.
Okay.
She's coming from the University of Brest. And we also have a second speaker, Wojciech Pokora, coming from the University of Gdańsk, from Poland. So I would like to say that Trinetta is a postdoctoral researcher at the BIP laboratory at the University of Brest. And Wojciech is already assistant professor at the Department of Physiology and Biotechnology at the University of Gdańsk. And we are going to start with Trinetta, that she's going to give a talk regarding blue growth. And she's working she's doing research with very special group of bacteria that support high pressure, and they live in very deep environments. And she's studying the potential for the biotechnological potential of this group of organisms. I think that she's an example of a researcher that does an important interdisciplinary research, since in order to achieve her objectives in research, she's using, or she's involved in microbiology, in genetics, in proteomics, in all kind of omics, and bioinformatics. So it's a very good example of interdisciplinary research. Today, she's going to talk about, I'm going to read the title of her talk, is Physophile Adaptation to Amino Acid Deficiency at High Hydrostatic Pressure Conditions. So we are, we are looking forward to hear more about this group of bacteria and what can they provide in sustainable blue growth. So I would like to give the floor to Trinetra, well, just let you know that you have a specific time for giving your talk, and then we will have a short section of questions before listening to our next speaker. So the floor is yours, Trinetra.
Thank you.
Thank you.
Can you see my screen?
Yes, we can see. So thank you very much for giving me this opportunity to present my research work. And as already I am already being introduced, so I am Bienvenue Marie Curie postdoctoral fellow working in the University of Brest. And as already mentioned, I work in the BEEP lab, which is the Laboratory of Biology and Ecology of Deep Sea Ecosystems. And it's a combined unit of UBO, means University of Brest, CNRS, and IFREMAR. And the project on which I'm working on is funded by Bienvenue Marie Curie Organization and Region Brittany under the Marie Curie Grant Agreement. And I'm working on protease production as a possible mechanism of polypeptide degradation for amino acid acquirement by deep sea piezophile under high pressure conditions. So first, I would like to give a brief introduction about these specific piezophilic environments. So what is high hydrostatic pressure? As we look at the ocean, there is an increase in pressure, hydrostatic pressure, at a rate of 10 megapascal per kilometer. So environments above 10 megapascal qualify as high hydrostatic pressure regions. So this is the region of high hydrostatic pressure. And this region is very important because it is about 88% of the ocean volume. So this region cannot be ignored. And it's a very important part of the ocean ecosystem. It's physiology, it's biology, and the geology and everything. It's very important to study. The average pressure in the ocean is around 38 megapascal, and it can reach up to 110 megapascal in deep trenches. So these are extreme environments. Extreme environments mean environments which are considered deemed to be extreme for living, and these piezophils live at pressure conditions which are greater than or equals to 10 megapascal. So piezophils are these kinds of extremophils which live at this environment. Now, even in this piezophilic environment, we are more focused on a specific type of piezophilic environment, which are called hydrothermal vents. Now, why are these hydrothermal vents unique areas? Because these areas are like a combination of different environmental conditions.
Now, these hydrothermal vents are formed due to the activity of tectonic plate rifts, and the hot basalt and magma, they cause it to drift apart. So what happens is that the seawater, which contains oxygen and various oxygenated compounds, and which has a low temperature of about 2 degrees, it seeps into the cracks. But once it seeps into the cracks, it mixes with different hot minerals, and then this fluid is emitted from the springs. So this hydrothermal fluid then contains different kinds of gases like hydrogen, methane, hydrogen sulfide, carbon dioxide, and it also contains heavy metals like manganese, iron, silicon, and zinc. And this is also anoxic in nature. So it means it does not contain oxygen, and it has acidic pH, and the temperature is very high. So it's greater than 350 degrees Celsius. So we can see in this small environment, there is a huge environmental gradient. And that's why it is a very unique hydrostatic pressure environment. Now, when we look at this environment, we can think that these environments are so extreme, and how can life sustain there? But I can tell you that there is life living in this hydrothermal vents, and not only microbes, but also annelids like tube worms. They also live in these environmental areas. So it supports and sustains a number of biological growth. And now I come to the piezophil, which I am studying. So it's called Thermococcus baerophilus, and it's a piezophil which was isolated from a similar kind of hydrothermal vent, which is called the snake pit hydrothermal vent, and it's located in the mid-Atlantic ridge. This is the picture of the hydrothermal vent. And from here, this organism, this is an archaea, and it was isolated. This is the electron micrograph of a cell of Thermococcus baerophilus. So it is the first true piezophilic hyperthermophilic, means it grows at very high temperatures, archaea isolated from the hydrothermal vent system. And interestingly, it not only sustains or it grows at 40 megapascal, but even its optimal growth is at 40 megapascal. So it means it needs this kind of hydrostatic pressure to grow optimally. And it also grows at 85 degrees Celsius and 3% salinity. But interestingly, it can also grow at atmospheric pressure, meaning 0.1 megapascal, which is its suboptimal pressure for growth, and also at higher hydrostatic pressures. So it can grow up to 70 megapascal, though the growth is not optimal at this pressure, but still we can see that it can grow at a high range of hydrostatic pressure. It has a circular chromosome of around 2 megabase and also a plasmid. Now, when we are talking about hydrostatic pressure, we need to see that why hydrostatic pressure is being deemed as an extreme environment. This is because hydrostatic pressure or high pressure can have various deleterious effects on a cell. For example, the cell membrane, which is generally semi-fluid in nature, it loses its fluidity and functionality, and it becomes rigid, so it no longer remains functional. The mobile elements like flagella, they can become non-mobile due to the pressure. And on the proteins which are folded, they can become unfolded. Its various multimeric subunits can be broken down into monomers, and various physiological processes are hampered within the cell. So if there is high hydrostatic pressure, and if we put a normal bacterial or archaeal cell or any cell within that pressure, all these kinds of things will occur, and this organism will not grow like that. But the interesting thing is the piezophils which are living at this environment, they have clearly devised a number of mechanisms to counteract these deleterious effects. For example, it regulates the overall gene expression. Like some genes are specifically expressed at high pressures. There is a fine-tuning of the transcriptome of the proteome, compensation of the loss of activity of some proteins, and last but not the least, there is also a biomolecular structural adaptation. So it adapts in a various kinds of ways, and this piezophilic adaptation is very interesting too, also because when it adapts not only to the high pressure, but as we saw, it has also high temperatures, high salinity, and low oxygen content. So it has to adapt to a number of environmental conditions, some of which might have opposite effects on each other, but still, interestingly, it adapts in all of these extreme environmental conditions and then can grow and flourish. So in this pesophilic adaptation in my project, I'm specifically interested on the genes which are specifically expressed at high pressures. And so I'm studying mainly this part and not the structure adaptation or the regulation. Now I come to the background of my research problem. So to make up as like a background, I would like to say that the genome and transcriptome of this organism, like Thermococcus baerophilus, was already sequenced when I wrote this project. And we got various interesting facts from this genome and transcriptome data. And specifically, I'm interested on the facts which are related to the amino acid biosynthetic pathways because this forms the crucifix of my project. Now I get to the genome data. So from the genome data, the amino acid biosynthetic pathways were deciphered and not all the pathways were found. So complete pathways were found for these 7 amino acids and incomplete pathways were found for the 6 amino acids. For the other amino acids, they were either not found or they were found only up to distant common precursors. So this was the data obtained from the genome sequence of Thermococcus baerophilus. Now, there was the transcriptome data too, and the transcriptome data formed the backbone of the research problem. So it was very interesting to note that there was downregulation of several known and hypothetical genes which are involved in biosynthetic pathways of various amino acids. And these amino acids were proline, threonine, arginine, aspartic acid, histidine, aromatic acid precursors, glutamine, glutamate cysteine, and glycine. So from this information, we get to know that these amino acids are synthesized in low amounts because there is downregulation of the genes producing these amino acids at high hydrostatic pressure. So this is one thing. And then there was another experiment carried out by a researcher in the University of Bordeaux, Kerio, and she carried out an experiment on the amino acid requirements of Thermococcus baerophilus as a function of growth pressure. What she did was she grew the organism on Thermococcus bacil media, giving all the salts and acetate using as a carbon source. And she supplemented with a mixture of 19 individual acids. So we know there are 20 amino acids. And in each case, she omitted one amino acid because she wanted to see the effect of the deletion of the removal of individual amino acids from the media. Now, when this experiment was performed, it was found that after 24 hours at low atmospheric pressure, the missing amino acids, their effect was not that much because the growth yield decrease was not larger than 0.3 log units. So there was not much decrease in the growth. However, at 40 megapascal at high hydrostatic pressure, at least 13 amino acids led to yield decreases larger than 0.36 log units. So it means that at high hydrostatic pressure, at least when we are not giving these 13 amino acids individually, the growth is much less. And this effect became more pronounced after 72 hours. So the decrease in growth yield became even more larger. So from this experiment, we can see that if at low atmospheric pressure, if we are giving amino acids from outside, if we are omitting the amino acids, then it does not have much effect. But at high hydrostatic pressure, it needs a number of amino acids to be supplemented from outside because clearly it cannot biosynthesize these amino acids within itself. So from here, we got a table regarding the amino acids which were essential in baerophilus and as a function of growth pressure. And it was found that only 3 amino acids, that is alanine, glutamine, and proline, they were not essential for growth at 40 megapascal. So it means that it needs all the other amino acids to be supplemented from outside for growth at high pressure, and it cannot biosynthesize these amino acids. However, at atmospheric pressure, it needed only asparagine, cysteine, and tyrosine for growth. So for the other amino acids, they could already synthesize or biosynthesize these amino acids within itself because its biosynthetic pathways were not depressed. Now we know that amino acids are very important building blocks of the body. They are essential for proteins, they're essential for hormones, and for various other structural and functional components of the cell. So here comes an essential survival question. Then how does this organism, Thermococcus baerophilus, obtain the necessary amino acids for growth? Because if it does not obtain the necessary amino acids for growth at high pressure, then how will it survive? To answer this question, we formulated a hypothesis. So our hypothesis is that probably it produces a unique array of enzymes, particularly proteases or peptidases, to degrade polypeptides, which are already found within its natural environment or outside. And then they might have dedicated membrane transporters by which they uptake the peptides. Now, testing this hypothesis is the main point of our project. So what are the potential outcomes of this project? Why this project is important? Because first, it helps in deciphering a basic mechanism of piezophile adaptation. And as I have already mentioned, that piezophilic environments are very important in case of the deep sea environments for studying how the organisms are surviving there and how they are flourishing there. So, and they are very important part of the deep sea ecosystem. And secondly, the characterization of a thermopressure stable protease, which can have biotechnological potential. Proteases have applications in different types of biotechnological industry, like food industry, dairy industry, peptide synthesis, detergent industry, and animal feed waste management. So there are many proteases which are commercially available proteases, but importantly, in industry, we require proteases which are very stable, like stable at high pressures, at high temperatures, because we need robust enzymes which are not degraded or inactivated at low pressures or low temperatures. So it is very important that we have some really thermopressure stable proteases having biotechnological potential. And proteases from piezophils have not been studied like that. And previously there has been the study of only one, like report of only one piezophilic proteases and not even detailed study. So with this respect also, it is very important, this project, the outcome of this project. So what are the objectives of the project? Firstly, the detection of proteases and uptake transporters, which are differentially expressed. Since we hypothesize that at high pressure, there will be proteases or transporters which are expressed either overexpressed or expressed uniquely at high pressure. So the detection of particularly these proteases which are expressed at high pressure. Then secondly, identification of those proteases which are particularly involved in amino acid feeding for survival. Thirdly, purification and structural characterization of the most important protease. So to do to test the hypothesis, we grew Thermococcus baerophilus at both low and high hydrostatic pressures at 85 degrees Celsius. At low pressure, it is relatively easy to grow, but for cultivation of microbes at high pressure, we need this specific high temperature, high pressure reactors. These are the reactors, and we put the cultures in small vials inside them, and then we regulate the temperature and the pressure with the help of this pressure valve. And this is very tricky to perform because these instruments are physically very exhaustive and they require high expertise, and also it's not easy to work with these instruments. So culturing them at high hydrostatic pressure is relatively more difficult than culturing at low pressures. Then once we got sufficient cultures, because it was done in a kind of scale up kind of way, first I started with small volume of cultures, then proceeded to large scale volumes. I extracted triplicates of proteome samples from different subcellular locations. The secretome are the proteins, the proteome pool, which is secreted by Thermococcus baerophilus outside of its cell. Lysate, which means the cytosolic proteins, the proteins which are present inside of the cell. And membrane proteins, proteins which are attached to the membrane part of Thermococcus bairophilus. So in total, there were 21 samples considering all the replicates and also controls. Next, I analyzed them in SDA's page analysis and also mass spectra to find out the high hydrostatic pressure expressed proteins and identify the particular proteases and transporters. So from the proteome study, we got various interesting results. For example, this is the SDS page profile pattern of the secretome, the lysates, and the membrane fraction. To explain this image, these are the gels from high pressure. And as I mentioned, it was grown in TRM, like thermococcus rich medium. So these are the proteins from high pressure from 40 megapascal. And this is the profile pattern from low pressure, that is 0.1 megapascal. This S represents the three replicates of secretome, secreted proteins. L represents the lysates, and P represents the membrane bound fraction. So we can clearly see that there are differences in the patterns. For example, there are additional bands at high pressure, and some of the bands, we can see that for some of the proteins, the bands are present at low pressure, but it is overexpressed at high pressure. So clearly there is a difference of expression, and also in the different samples like secretome, lysate, and the membrane bound fractions, they have a number of differences, and we can see there are a large number of proteins. So it was not possible to predict the proteins from this SDA's page profile, but it was possible to see that really there are differences. So there is differential expression at low pressure and at high hydrostatic pressures, as we can see. Then once we got the SDA's page profile pattern and the proteomes at high volumes enough to be sent for mass spectra analysis, they were sent to the EDYP platform in Grenoble for mass spectra analysis. From the results of the mass spectra analysis, we could identify a number of proteins which are differentially expressed at high pressure and low pressure. So we can see here these violet ones, or these violet dots, they represent the proteins which are expressed or which are found in high abundance at high pressure. These green dots represent the proteins which are found in high abundance at low pressure. And these gray dots are the proteins which are neutrally expressed. So it means there is no difference in expression of the proteins at high pressure or low pressure. And we considered only those proteins which had a fold change value of 2 log, and proteins which had a fold change value less than 2 log, they were not considered. So as we can see, there are a number of proteins which are found in high abundance, either at high pressure or low pressure. In the case of secretome, in lysates, and the membrane bound fraction as well. Here we can see the different percentages of the proteins. So in the secretome, a large fraction of the proteins, they are not differential. And this is true in case of not only secretome, but also the lysates and the membrane bound fraction. So a large number of proteins are neutral, but the good news is that a large number of proteins, they are differential at high pressure and at low pressure. So 26% of the proteome, this is expressed differentially at low pressure, at high pressure, and 18% of the proteins are expressed differentially at low pressure. So this is in case of the lysates. Also 8% of the proteins, they're expressed highly at high pressure, and 6% of the proteins, they're expressed highly at low pressure. And for the membrane bound fraction, the percentage was almost the same. So we can see there is a lot of difference, and there is a differential abundance of the proteins as evident from the proteome study. Since we are more interested about the proteins which are found in high abundance at high pressure, as we need to study those proteases and uptake transporters which are expressed highly at high pressure, we filtered to those proteins only. So in the secretome, there were 159 proteins which were found in high abundance at high pressure. In the lysate, there were 118, and in the membrane-bound fraction, there were 37. And there were several overlaps between these proteins. So some proteins which were found in the secretome are expressed highly in the secretome. They're also found in high abundance at the lysate and the membrane-bound. So since we had a large number of proteins and we do not need to study all the proteins. They will be used for another study, all this whole proteome study. We are more interested about the proteases. So we selected some of the proteins. Now, you may think that why I am selecting hypothetical proteins too. It was very interesting to note that most of this secretome proteins, that is proteins which are highly expressed at high pressure, the best ones or the best hits, they were hypothetical proteins. So there is a probability that there might be unique proteases which the domains which still have not been studied. So probably we can find some unique proteases or proteins among the hypothetical proteins, although it is difficult to decipher the activity or the functionality of all hypothetical proteins. And also I have not given this data here, but for some hypothetical proteins, a detailed study has been performed, and for some, there are some protease-related domains. So, and 13 were already proteases or peptidases which were highly expressed at high pressure in the secretome, and 6 were uptake transporters. And these are the data from the lysates and the membrane. So in total, initially, there were 105 target proteins. But since we are more interested about the secretome, so we set to work on the proteins found in the secretome, that is the 57 proteins consisting of hypothetical proteins, proteases, and transporters for further study. So once we had this 57 proteins, we performed a detailed in silico study of the target proteins, and I used a number of bioinformatical softwares and tools to study these proteins, particularly the hypothetical proteins, as it was very important to study these hypothetical proteins in details before concluding that they really have some protease domains or not. So we performed physiochemical analysis of these proteins, the abundance analysis at different pressures, a comparative analysis of the proteins as compared to other proteases, seeing the interacting proteins, especially for the hypothetical ones and also for the proteases, whether they are unique proteins or not, which proteins they are interacting, the subcellular location, because we are concerned about secreted proteins, and the subcellular location should be outside of the cell, not inside of the cell. So it was very important to predict or study whether the proteins are really from the outside of the cell or not. Then the sequence similarity analysis and phylogenetics. The flanking gene study, that is the genes which are located at the upstream and the downstream, and also the conservation of the orthologous, the homologous genes. And finally, the comparative structural analysis, especially for the hypothetical proteins to find out the probable function of the proteins. So after this extensive study was carried on, we selected some of the proteins because it's not possible to work on all the 57 proteins. And so we selected 5 or 6 targets, like since we worked on all the proteins. These are the predicted structures of some of the protein targets, and these were obtained with the help of in silico analysis by either AlphaFold or Rosetta Fold. So as I already mentioned that after this analysis, we selected 4 or 5 target genes, and currently only these genes are being focused on. So the ongoing work is that the final 4 or 5, the 5 target genes which have been finalized, corresponding to the detected proteases and transporters, they have been selected. And now I am constructing the corresponding gene knockout mutants, that is the genetic engineering part. I'm doing this right now to knock out the particular genes against those proteases which I think are interesting targets and might have a role. And once I am successful in constructing all the gene knockout mutants for the corresponding 5 genes, I will observe the effect of the gene deletion of each gene on the growth of Thermococcus verophilus at high hydrostatic pressure, because I need to see whether really these are the proteases which have an effect on its growth, and also on the amino acid uptake of Thermococcus verophilus, because it's important to know whether that protease is really important, that is really secreted at high pressure to counteract for the amino acid biosynthetic deficiency. And once the genes are selected, like we find out which genes are responsible for this amino acid uptake, the functionally most important protease will be purified and characterized. And this has been envisaged like a secondment at the University of Grenoble. I am thankful to my supervisor, Professor Mohamed Jebar from BEEP, for hosting me, Dr. Sebastian Laurent, who has been instrumental in helping me and guiding me for the proteome analysis, Dr. Jan Maulik from Eisen, again in Brest. He has been helping me with the genetic experiments, Dr. Bruno Franzetti, our collaborator in the University of Grenoble, all the other BEEP lab members who are helping me a lot to progress this project. And importantly, I'm also thankful to Bienvenue Marie Curie Fellowship and Regine Brittany for funding my project. Thank you very much.
Thank you very much. Sorry. Thank you very much, Trinetra. Thank you very much for your presentation. I think that it was very complete and very well structured and really interesting. I have some questions, but as the presentation extended a little bit more than what we expected. I think that we leave the questions for your research to later on, if you don't mind. And I would like to give the floor, or I would like first to present our next speaker. Is that all right?
Yes, sure.
Okay. So would you mind to stop sharing?
Yes.
Thank you. Thank you very much. Very nice presentation. Very interesting. Okay. So I would like to introduce our next speaker, who is Wojciech
Pokora. Wojciech Pokora.
That's it. He's, as I said before, he's assistant professor at the University of Gdańsk, and he studies cell cycle progression and regulation, and how is this being affected by exposure to nitrogen oxidized or hydrogen peroxide, and more concretely in cells, in single cell platonic algae. I mean, these important organisms which are the base of the trophic of the trophic change in our seas and in our oceans. So I'm looking forward to listening to you to find out how these substances can make a difference in the regulation and in the progression of these important organisms. So I give you the floor. With the presentation, which is entitled Hydrogen Peroxide as a Tool for Green Algae Growth Regulation, how it works. The floor is yours.
Okay, thank you very much. Thank you very much. I have a possibility to present the things I'm working on in the University of Gdańsk. Maybe I'm not a very young talent in here in this area, but I still feel like a young researcher. I'm generally an experimental biologist, and I work mostly in laboratory, but I think that the scientific things that I'm working on are quite well linked with the blue culture, especially 2 years ago. I had the opportunity to visit the University of Brest. So then we have a visit in the scallops farm where they were fed with green algae, and I think the methods I'm working on could be very very useful for such culture or such analysis. Okay, maybe I will start from the some of things that I used to do. Generally, I was interested in physiology of stress. I was particularly interested in oxidative stress in green algae. The stress was induced by two groups of pollutants, the heavy metals and herbicides. I was focused on the substances that were put into the environment, especially water environment, by humans. So this is why two of them, but we are still in the subject of the stress. In the previous presentation, we had let's say abiotic physical stress, here typically chemical stress. I was interested in many overreactive oxygen species. Particularly, one of the most interested for me was hydrogen peroxide, because besides the fact that it is toxic for in many cases, for some reasons it's quite useful and acting as a signaling molecule in many aspects. It's proven in green algae, proven in higher plants, also in humans or in animals. So I think that this part of reactive oxygen species action is quite universal. In general, we have two aspects of action of hydrogen peroxide. We have something that we call redox biology, and there is a lot of reactions in the cell that is regulated by hydrogen peroxide or other reactive oxygen species. They are generated during peroxisomal activation, mitochondrial activity, chloroplast activity, and so on. They also play a very important role during transportation of the electrons and so on. And the production of hydrogen peroxide is interested because this is one of the molecules that has possibility to move through the cellular membranes. Some of the others, like oxygen ion, are not able to move through the membranes, and this molecule is quite long-living and quite long distance acting. Of course, it's possible to modify it is possible that it modify structure, and especially sulfur amino acids, sulfur-containing amino acids in proteins, and this seems to be this most important signaling function. It works in gene expression, in development, in cell cycle progression, and so on. Of course, on the other hand, we have oxidative damage according to a lot of oxygen species, like DNA damage, protein oxidation, lipid peroxidation, cross-linking, and so on. But here there's also a lot of mechanism that prevents cell from this damages, like antioxidative enzymes or low molecular antioxidants. I was mainly focused on the antioxidative enzymes, because I'm the one that culture algae, unicellular algae. So I have two methods for culturing them. One is asynchronous culture, and this is the very typical for most of the cultures. When the growth of algae is not controlled, they grow as they want to. Some of them are very young autospores or zoospores. It depends if they are movable or not. Some of them are just in the phase of growth, and some In some of them, the growth is terminated and they're just upon to division to young daughter cells. In such culture, it reflects in many ways the natural environment, but when we want to analyze the particular enzymes or what happens during the cell cycle, it's quite unuseful, and that is why I use also synchronous culture here by application of the some very favorable conditions like high CO2, optimal light intensity, and the change of the light, the photoperiod. It depends on the species, but usually it's 12 hours of day and 12 hours of night. We can somehow make the algae to grow in synchronous way. So all the cells in this culture start to grow, then the growth, and in the same moment, in similar moment, all them start to divide. That's something like very disciplinary growth in the army, I would say. In this case, there is one great thing about this type of culture. I can work on the large population of the algae, but interpretation of my results is on the level of a single cell, because even if I analyze 100, 1,000, 10 million of the cells, they are unified and they are in the same moment of their cell cycle. This is the advantage of this culture. My analysis, I started from the asynchronous culture because I was interested in finding out what is the difference between some strains are sensitive to chemicals and some of them are not. I was analyzed a lot of species. Most of them are isolated from the Baltic Sea, from our coastal zone of the Baltic Sea. This Desmodesmus armatus and Desmodesmus subspicatus or obliquus. This green algae may be well known to you because it's used also in the ISO standard toxicological test. The conditions of this test are quite different because then the medium is very low concentrated, the growth of algae is very slow, and the number of the cells is also very little. I had to adjust it to the biological and biochemical analysis. I had analyzed parameters like growth of the population, efficiency of photosynthesis, activity of antioxidative enzymes, and I had selected strains that reflect very different sensitivity to example of heavy metal. I was working with cadmium, and two strains that are that expressed very different sensitivity to chloridazone. It's a herbicide very commonly used in European Union. Then, after knowing that these strains may differ instantly, I choose my growth model for a cell cycle study. Here you have the general overview of such a culture. The cell starts to grow, and then we have one division, second division, and the third division usually. It's some kind of biological adaptation of green algal cells to the environment, because in many cases they divide only asexually, only by mitotic division. So after passing something that we called commitment point, the cell do not finish the first cycle, the first round of the cell cycle, but start the next one that overlap the first one. This is something that is happened on the light, and the termination of the cell cycle, the one round, second round, or third usually, is restricted to the dark side, because first of all, these algae use solar light as long as it is possible. And due to the fact that they divide in every 24 hours or not very, or 48 hours, the synthesis phase occurs in the night when there is no UV radiation. So the opportunity to make a mistake during the synthesis of DNA is much lower than when it happens on the light. This is how it looks in the microscopic version. Small zoospores, then it grows, and finally we have mother cells with usually 4 or 8 daughter cells in the end. In this diagram, you can see what happens in two Desmodesmus strains that are sensitive and resistant to cadmium. During the cell cycle, here we have the version of the cell cycle, the cell grow, and here we have division to the daughter cells. Of course, it's usually take photosynthesis. The arrows down indicates that there is inhibition of photosynthesis, very important. Because we work on the cadmium, so we analyzed also some specific proteins that are involved in adaptation to cadmium stress, like phytocalatins or HSP proteins, but we saw a very important differences. For example, in the resistant strain, there was a much higher synthesis of phytocalatins or HSP proteins, and the photosynthesis was not so much inhibited. What was most interested for us was the changes in number of proteins and the activity of antioxidative enzymes like superoxide dismutase, ascorbate peroxidase, or catalase. In this case, the resistant strain exhibited much higher amount of these proteins and much higher activity of these proteins. That this was not very surprising for us. In this case, the cell cycle was not very affected. The cell was able to finish this cell cycle, and there was no much difference between the control culture and the culture of resistant strain. Some much more interested results we had when we went to other version, when we induced the stress with herbicide. Here we have chloridazone, the same culture system. We also have 2 strains. One of them is sensitive and the second one is resistant. In sensitive strains, there was nothing very surprising. Photosynthesis was inhibited. The analysis of chlorophyll fluorescence indicated that many of the photosynthesis PS2 centers were switched off and they are converted to the heat sinks. So this was the main reason why the photosynthesis was so inhibited. A lot of reactive oxygen species were detected, and there was no changes in the amount of antioxidative proteins, and their activity was also not affected. And as a result, we could see the inhibition of cellular growth. Most interesting things happened when we started to analyze the resistant strain, because the dose of herbicide was the same, and the final result was stimulation of growth. We observed much more daughter cells released from one mother cells that in the control culture. First we thought that it was a kind of mistake or something, that there's something wrong with it, but the phenomenon of hormesis was detected previously, so I think maybe it was something like that. We start to analyze physiological parameters, and first of all, after a few hours of photosynthesis decrease, photosynthesis went to equilibrium, and in the end of the cell cycle, intensity of photosynthesis, which was much higher than in the control culture. We also observed a lot of active PS2 centers. So the efficiency of oxygen production and the efficiency of growth in this case was much higher. Also, we saw the resistance to herbicides in the amount and activity of antioxidative enzymes. Maybe the synthesis of these enzymes was not very high, but their activity was much, much higher than in the control cells. And that is why the amount of reactive oxygen species was quite on the quite low level. So in this case, we thought that containing the equilibrium of reactive oxygen species can work with the regulation of the cell cycle. And this was the something that induced us to work more properly on the analysis of how the cell cycle of unicellular green algae is regulated, and what is the actual mechanism. These two results was most important for us: the stimulation of photosynthesis and stimulation of growth in the end of the cell cycle. So in this case, we had to change the organism, because we wanted also to analyze this on the genetical level, not only a physiological level. So from Desmodesmus, we changed the model to Chlamydomonas, but we still have some questions that we wanted to ask. We were wondering if it is possible that hydrogen peroxide can regulate or take a role in the regulation of the cell cycle of microalgae. The cell cycle was parameterized by expression of cyclins and cyclin-dependent kinases. So we know what directly when we have the switch from the phase of growth to the phase of synthesis and division and so on. Photosynthesis efficiency, it was analyzed mainly by analysis of fluorescence of chlorophyll, homeostasis of reactive oxygen species, and here we added also nitrogen species, direct detection of hydrogen peroxide and nitric oxide, expression of enzymes involved in formation of hydrogen peroxide and nitric oxide. We did it on the level of transcription, translation, and the activity. And here, this is something I thought in the beginning, that we had to change the organism, because Chlamydomonas reinhardti is fully, we know the full sequence of genome. In case of Desmodesmus, the sequence, we know some parts of the genetic sequence, but not whole, not everything. Why we were interested in hydrogen peroxide, but also in nitric oxide? Because in many cases, there is a great crosstalk between these two molecules. Of course, they can act independently. Hydrogen peroxide may act by carbonylation of proteins, may translocate to nucleus and change gene expressions. The same activity is for nitric oxide. This also can act as a nitrosylative agent for proteins, translocate to nucleus and interact, for example, with zinc fingers, motif, or inactivate transcription repressors. But in many cases, both these molecules interact with signaling pathways. GMP, MAP kinase, cadmium, calcium efflux, and so on. So both of them are possible. In both cases, it's possible to modify signal transduction in the cells and modify transcription of some factors. Okay. Here we have a model of progress of the cell cycle in Chlamydomonas reinhardti. We start with the turning of the light. The cells start to grow. After about 8 hours, between the 8th and the 9th hours, we have the switch between G and S phase. 1 hour later, we have the switch between synthesis and mitosis starts. After 10 hours of light, we have a mother cell with 8 daughter cells inside. In the 14th hour, more or less, all the zoospores are liberated and they are ready to start the new cell cycle. We were interested in the physiological parameters, of course, photosynthesis. To make it clear, this is a situation with no stressors. This is just control culture. We kept this algae in very optimal conditions with no cadmium, no herbicide, optimal light, and so on. And this is what happened during typical cell cycle in the control cells. The photosynthetic apparatus developed, photosynthesis becomes more and more intensive, so more oxygen is produced. Because we are interested in hydrogen peroxide, we analyzed the enzymes related to its production and scavenging, like superoxide dismutase, ascorbate peroxidase, and catalase. They are highly active in the beginning. It's most probably up to turning off the light at the stress, physiological, I would say, stress that was induced by the turning off the light. And then the activity was more or less not very high. When you look at the level of hydrogen peroxide, we have one efflux of hydrogen peroxide in the beginning of the cell cycle. Then the level decrease. Then we have some changes in the amount of this molecule during the passage through G and S phase and S and M phase. Also, the beginning of the zoospore release is correlated with high efflux of hydrogen peroxide. The other molecule that was interested for us was nitric oxide, because, as I said before, they can interact each other. In Chlamydomonas ringhardi, nitrate reductase and nitric reductase are two main enzymes involved in production of this molecule. So we analyze their expression and activity. And this is how the changes of the nitric oxide production during the cell cycle looks like. What was very surprising for us, in some moments of the cell cycle, the production of hydrogen peroxide and nitric oxide were the regulation occur in the same moment. For example, when you have key moments of the cell cycle, passage through the from phase of growth to the phase of synthesis, amount of hydrogen peroxide increase, amount of nitric oxide decreases. In the passage in SM phase, situation was similar but opposite. One of the gases increased, the second one decreased. That was one of the results that let us know that there is possible that these two molecules, the levels of these two molecules, take a role in the regulation of the cell cycle. We wanted to prove it, so we choose 4 moments that we thought was interesting for us. The beginning of the cell cycle, the moment when the photosynthesis is very efficient, and 2 key moments of the cell cycle, beginning of the synthesis phase and beginning of the mitosis. The question we asked was if the change of intracellular ratio of hydrogen peroxide and nitric oxide can modify the cell cycle. So we choose the moment for the application, exogenous application of hydrogen peroxide. We made a map of changes that occurred during this activity, and we also found how the reproductive pattern of Chlamydomonas cells changed. We decided to change the level of hydrogen peroxide by the 50%. So look at the level of the first number here is also it's always the actual amount of hydrogen peroxide present in the cell, and the second number number is the value that we changed after our application. It is just application of hydrogen peroxide to the culture, nothing else, and then verification of the level. We decided to change it by the 50%. So from 100%, we have 150% of the amount recorded in the control cell. These are values in the femtomoles per single cell. And then after such application, the cell cycle was continued. And what happens? When we did some such modification in the beginning of the cell cycle, nothing has changed. Maybe the adaptation for the beginning of the light phase was a bit, let's say, lighter, and the release of hydrogen peroxide was not very high, but we still have 3 replication rounds in one cell cycle, and the pattern of the changes of hydrogen peroxide and nitric oxide are quite similar. Interesting things start to be observed when we did some modification in the 3rd hour of the cell cycle. It's the moment when the photosynthesis is quite efficient. One of the things that we had observed was a 50% much efficient transfer of electrons between photosystem 2 and photosystem 1. And that is why we think that the cell was able to utilize light energy much more efficient. The amount of energy was the same, that just the utilization was much higher. That results in faster growth, and until the cell cycle was terminated, the cell was able to pass not by 3 but 4 rounds of the cell cycle. So in the end of the culture, we have not 8 but 12 or more daughter cells released from one mother cells. We think we expected 16 because it should be 2 times more than 8, but I think that not all the cells were the same, express the same level of sensitivity. When such modification of hydrogen peroxide level was made in the 6th hour of cell cycle, the 6th hour of light period, we also observed the effect of photosynthesis efficiency, but because the time that left to the end of the light period was quite short, we think that these cells were not able to pass through the 4th round of the cell cycle, but volume and biomass of these cells was much higher. When you look here for the average volume in the control cells was about 80 micrometers cubic. Here we have 105 micrometers cubic, so it's quite much more efficient growth. And the 4th results maybe not so spectacular. Application of hydrogen peroxide at the end of the cell cycle allowed to earlier liberation of daughter cells from the mother cells. It was quite interesting because we did such experiments on the cultures when the liberation of daughter cells was inhibited, and this application of hydrogen peroxide also overpassed this inhibition version. And this is something that we had already proven, and we know that works. It has been already published, but we know that it is possible to accelerate the growth of synchronous culture by simple application of hydrogen peroxide. It's a physiological molecule, so this is nothing very chemical that can be used, or not a fertilizer that can be added to the culture. What we are working now, it's in the present study, we are interested in the mechanism, in identification of the mechanism, how it works. first area is post-translational modifications of the thiol residues in cysteines.
I'm sorry for interrupt you, but we are running a little bit out of time.
Okay.
So, yes.
Okay, so two slides more. Direct regulation of the cell cycle. We have identified a lot of molecules that have exposed residues, cysteine residues in Arabidopsis and in Chlamydomonas. And the second area of our research is analysis of ferredoxin as key coordinator of the Calvin-Benson cycle, because we think that this regulation is also possible on the not only on this light-dependent reaction, but also during on the Calvin-Benson cycle. Okay, thank you very much, and sorry for prolonging a bit the time.
Thank you very much. You know, we're running a little bit out of time with the first talk and the second one, but I found it so interesting that I didn't want to interrupt. And,
yeah, of course, no problem.
So thank you so much. I found it very interesting. And regarding the presentation, I found your figures very illustrative. I mean, easily showing what is going on with these cycles, with all the enzymatic activities, how they are changing over time, depending on the growth of the algae. I found a very interesting piece of research, since, I mean, it's a way to increase the growth of algae that maybe they can be used for, I don't know, for production or for decoration of residual water treatment plants. So I found it a very important application in the research that you have been presenting today. I have to say that I'm working, maybe I will contact you after summer holidays, because I'm working with Cymboridinium in nidarians, and we are measuring biomarkers, oxidative stress, lipid peroxidation, DNA damage, and we are also there were things there that made me think, because we have, depending on the stress, because of temperature, we have like different concentrations of cells and size of the cells. So, and it's something that we couldn't interpret. So maybe I will contact you after summer holidays.
Of course, there is no problem. With pleasure, we can either meet on the Zoom or Teams, or according to the CU meetings, we can visit each other. So of course there is no problem. I will then show you our culture methods from cell cycle analysis.
Okay, thank you very much. So I will open like a 5 minutes question period, because we are a little bit out of time. So are there any questions? Is there anyone? Well, I mean,
if it's possible, maybe one, maybe not question, but comment to Trinetta, if it's possible.
Yes,
I think it would be great if you will be able to contact with a head of our microbiology department, with Professor Kaczorowski. I will write you in the chat in a moment his name and the faculty, because it will be easier, I think. He also, I'm plant physiologist, not microbiologist, but I know that Tadeusz is also working with extremophiles in some other version, because he works mainly with exthermophiles that grow in thermal high temperature conditions, and he's working on polymerases. But I think, as I know, he wants to start working on the marine bacteria also. So maybe there will be a lot of experience from you to our faculty, if it's possible.
Or maybe also the reverse.
It's I think that is why we meet here, so it will be much more quite fruitful, I think. Okay, just chat. I have to.
Okay, thank you very much. I had some, regarding Trinata, I had a question, because I always, this is something that I have had in mind many times, which is to simulate deep sea conditions in the laboratory. And I saw that you are using that as special infrastructure, special instruments to simulate pressure in organisms. Do you think this infrastructure, this could be used for larger organisms? I'm talking, for example, about invertebrate species, for example.
At least our instrument, with respect to that, I don't think it's possible, because what we do is we culture the microbes in small vials, and in a media which has all the necessary salt concentrations to simulate the seawater. And then we put it in the reactor, which I showed, and then we fill the reactor with water. So the vials, the glass vials, they are like 15 ml to 25 ml. The maximum capacity of vial which we can put inside is 25 ml cultures. So we put them inside and we add water, and then we pump more water to stimulate the hydrostatic pressure. So for culturing even like bigger animals, we need bigger incubators.
Okay, and you take the sediments, the deep sea sediments, you sample the sediments in water.
For the isolation, this organism was already isolated before I came into the lab. So in IFREMAR and UBO, they organize some cruise, like scientific expeditions after every few years. And then they go to these hydrothermal vents and they collect the samples, and there are like specific instruments which go deep inside the ocean, and they pick up like water, sediments, and everything.
And then back to normal atmosphere.
But I have not collected. Maybe sometime in future I will go into some cruise like that.
Well, it's good to know that you keep it in the lab, and because, as you said, they can manage well at normal atmospheric pressure. So, well,
how they are still alive after changing of such pressure, because I was always wondering what happens during the change of the pressure. I think they are somehow able to adapt to the increased pressure, but after you turn it off, are they, I don't know, are they still alive?
Yeah, well, the thing is, it's like when we are taking out the vials, it's really important to take care that we are not reducing the pressure suddenly, because in that case, all my cells will die, and which has happened previously when I was first learning. So I was culturing them. When I'm taking them out and I see, okay, there are no cells, all of them are dead. So we have to decrease the pressure, like release the pressure in a really very slow manner, so that the pressure change is not sudden, it's gradual. And that is the good thing about Barophilus, that it is not an obligate piezophile, because there are obligate piezophils which can grow at only high pressures. But if we work on obligate piezophils, we cannot compare the two pressure conditions. So this is like a facultative piezophile, since it can grow at low pressure as well. That's why we are able to compare at the two pressure conditions. But at high pressure, it grows better. So at low pressure, it grows less. But that is the thing, like they adapt somehow. But even if like, sometimes there is some unexplained causes, maybe the pressure change, like it has to be really slow, and maybe it was not as slow as I thought. But then I take out the cells and I see they are broken. And also sometimes in the experiments, because there are many technical constraints to using this kind of instrument. Sometimes my vials break due to the hydrostatic pressure or the rubber stop cocks, they just pop out. So I have to do it again.
Hmm.
So in that case, the cells die.
Hmm.
Yeah.
Okay, very interesting. So thank you very much for participating as speakers in this webinar. I'm very grateful for your time and for your effort of preparing the presentation and being here today. And I will also like to thank the people attending the webinar. As you know, you are going to be able to watch this webinar in the YouTube channel, and also it's going to be saved and kept in the CU Academy platform of the CU Alliance. So thank you very much. I don't know if Maria would like to say any words.
I would like to join with your words, Laura. I think thank you so much for participating, for speakers, for Laura. Thank you for leading this very interesting webinar. And I would like to add also that the certificates of the webinar will be sent in September to all participants and speakers. And if you have any questions, you can ask us or send us an email, and we will stay in touch. So thank you so much.
Thank you so much. You can follow us in Facebook, LinkedIn, and Instagram. In Instagram, we are publishing many different our deliverables, the deliverables of the project, but also we are disseminating research projects. So if you're interested, you can contact us and we would be able to disseminate more the research that you are performing. So thank you so much. I don't know if you already took your holidays time or if you are going to take it in due time, but in any case, I wish you a peaceful rest and enjoy a little bit summertime. Thank you very much.
Thank you very much.
Thank you
Marcelo.
very much.
Thank you. Bye.
Thanks. See you. Bye.
Bye.